All right, second part of Unit 6 on animal form and function. The first part I want to re-mention is gametogenesis, the forming of gametes. With guys, you're going to have a diploid cell, a 2N cell, that's going to eventually split and hit meiosis. You're going to split into 2 haploid, 4 haploids, and then they're going to differentiate, get the whole tail spinny deal. With our females, when it goes through meiosis, you get what are called polar bodies, where almost all the contents of the cell goes to one side and not the other. Ultimately, what's left after the polar bodies is an egg, and that egg can be fertilized. Once that happens, you are left with a fertilized egg that hits different stages, 4-cell stage, blastula. It's going to start to actually form a hollow inside to it. That you end up with something like this if you look at it from the cross perspective. And then from there, things are going to start to differentiate instead of just straight up embryonic stem cells. It can become anything, what are called totipotent, into pluripotent, where they can become a few things. So on this, blue is represented by ectoderm, basically things that are going to be toward the outside of an organism. Yellow is endoderm, the inside, and red is the mesoderm, the middle. And it starts to fold in and either stays folded in and has one opening, or makes its way to the other side and makes a mouth. That's why at one point we were just a butthole, but then we got a mouth. As far as what do they become, ectoderm's the outer stuff, epidermis the skin, nervous, pituitary, jaws and teeth, germ cells. Mesoderm's middle, skeletal, circulatory, muscular, adrenal cortex, and then endoderm, a digestive tract, and the inner linings of some other tracts, as well as some glands. As far as prevention of fertilization, I'm just going to point this out. Very high failure rate on the old coitus interruptus. This is the first part. This is the second part, which leads me to probably my favorite sentence in the book. Also, a split-second lapse in timing or willpower can result in millions of sperm being transferred before withdrawal. Pull-out gain not strong. Moving on to a nervous cell, neuron. This is actually 2 neurons together, where the signal is coming in this one. This is called presynaptic, postsynaptic, assuming that we're talking about this synapse. Okay, so signals coming in, a stimulus comes in, it enters the dendrites, it goes into the cell body, it stimulates the axon hillock, goes down the axon, gets to the ends of the axon, what are called synaptic terminals, and in the synapse, that signal is going to cause neurotransmitters to release, which make it to the next cell, which then causes the signal to start in that cell, the postsynaptic cell, and then moves down that way. Nervous cells in general are either sensory integrative or or motor. Sensory is stuff that's coming in. Integration is sort of the middle of the road. Okay, the incoming information says this. What are we going to do about it? What we're going to do about it is the outgoing stuff, motor output. A lot of times that is movement, muscle. You can think motor is movement, or it could be gland stuff, excretions. I like these pages. I think it's worth taking a look at because it starts to tie things together. How a neuron works, do I think you need to know every little piece of this? I don't. I really don't. I think you need to understand that the charges inside the cell and outside the cell are different. Why are they different? Because of sodium and potassium. And sometimes sodium's let in, sometimes it's pumped out. Sometimes potassium's let out, sometimes it's pumped in, that kind of stuff. It's movement based on whether these gates are open or closed. And that movement causes different differences in charge from inside to outside, which causes that signal to happen. So the signal starts here, and it just moves its way down as sodium is going to flow in, flow in, flow in, and behind that, potassium is going to get pushed out, and you get repolarized. Neurotransmitters, again, that information's the signal is going to get here. Neurotransmitters are going to be released. They're going to bind to something else. When it binds to something else, that's going to cause flow of ions, which is going to cause the signal to start in the next one. As far as nervous systems in other organisms, you start with the cnidarians. As far as the nervous system, they're very, very simple. They just have a nerve net where all the nerves are interconnected with no really centralized location. Get into the flatworms, they have the first thing that we would consider a brain, two long longitudinal nerve cords, kind of like two spinal cords. They have an eye spot, so they can at least sense where information is going to and from. I don't know if I've been recording this whole time. Have I been recording? Your insects now have a brain, now a single nerve cord, and then ganglia, groups of segmented, or groups of concentrated nerve cells. And then we get into a salamander, a vertebrate, a brain, a spinal cord, definite ganglia, which are groups of neurons in one location. I like this picture because it shows central nervous system compared to peripheral, and it shows all the different types of glia. Glia, or glial cells, are helper cells. Neurons are so specialized that they can't survive on their own. They need all these helper cells to help them out. You need cells that help move nutrients in and wastes out. You need cells that help make coding to help speed up these nerves. These ones that are coded, that signal can travel faster. So you have something that causes a stimulus, and it could be temperature, pressure, whatever, light. That causes a receptor to sense it. It goes through afferent neurons going to the central nervous system. And then that processes that, sends out efferent going outward, and that can either be autonomic, think automatic when you see that, or it can be sort of manually, consciously moving things. So, oops, this is our conscious movements. We can consciously move skeletal muscle. Autonomically, think fight or flight, getting scared, getting angry. All of a sudden you get this rush of adrenaline, and then you also have to sort of bring that adrenaline back down. I think it would be worth looking through functions of the cerebrum, higher level thinking, left and right side of body, that kind of stuff. Cerebellum, coordination, diencephalon. You've got processing, sort of a sense, or where's information is going in and out. Hypothalamus, thermoregulation, biological clock, hunger, thirst, fight or flight, a lot of that stuff. And then brainstem, more survival-ish stuff. Think of this as a lizard brain, just enough to make you breathe and move and that kind of stuff. The higher level of the brain, the more complex the behavior that's possible. As far as a stimulus, you could actually be pushing on a neuron itself. That can be a way to send a stimulus, like a pressure. Or a stimulus can cause a cell to release neurotransmitters that go to a neuron. Muscle control, so Neurons connect to muscles. Muscles are made up of fibers and cells, and those cells have alternating patterns of protein filaments. Thick one's called myosin, thin one's called actin. One of these things is called a sarcomere, and they can pull together and then they can relax and spring back apart, and that's basically how muscles move. It's what's called the sliding filament theory. Again, do you need to know this everything just perfect? No, you don't. I think it's important to know that these little things kind of pull in on the thin ones, the thick ones pull on the thin ones, and we also need energy in order for it to release. If you don't have energy in the form of ATP, it can't release. That's why when somebody dies, they go through rigor mortis where everything tightens up. They cannot release. They don't have the energy release. Plus, their cells start to break down and release calcium. You've got two proteins on here, troponin, tropomyosin. One allows the head to attach, one doesn't. It's calcium that decides whether they're blocked, so whether myosin's blocked by tropomyosin or whether it's exposed through troponin. So tying it all together, you've got a signal coming from a nerve, releases a neurotransmitter, goes across the synapse, attaches to some sort of a receptor that causes that signal to move. It can cause the signal to move in a way that release calcium from the sarcoplasmic reticulum, endoplasmic reticulum. That calcium moves this out of the way so that this can attach, and it pulls, and ATP releases it, and then it goes back. And if we keep doing this over and over, we keep having movement, muscle contraction. Acetylcholine is the neurotransmitter here. Then you also need something to sort of break it down. So acetylcholine esterase, which takes it away and can allow you to start over. In ways that muscles are used, it depends on the organism. So this cartoon of my arm shows that, you know, you have contraction and you don't really have everything's a pull. There's really no push. Here the biceps is pulling, the triceps is relaxed, the biceps is relaxed, the triceps are pulling. You can apply this to a grasshopper as well. We've got a muscle that's flexing, bringing the leg in. We got the other muscle now flexing, pushing it out. Not really pushing it out, pulling it out. With things like segmented worms, you have a hydroskeleton. It's not a bone or exoskeleton. It's fluid, and muscle pushing and pulling against these fluid tubes allows them to move. And I think that's all we got.